So now that we've talked a little bit about measuring volumes with different types of pipettes, let's move on to talking about some of the assays that we'll be doing during this week. So during week 3, we're going to be doing the Bradford assay. Some of the principles of the Bradford assay is that it uses this Bradford reagent. It turns blue when it's reacting with any protein. So the Bradford assay is a really good measurement of the total number of proteins in your sample, the total concentration of protein. It doesn't tell you which proteins they are, or if your proteins are pure, or if it's a mixture of proteins. It just tells you, it measures pretty much almost all proteins fairly equivalently to one another. So for that reason, the degree of the color change is going to be proportional to the concentration of the protein that's present. So the Bradford reagent, when there is no protein, looks brown. And then the more protein that is in that Bradford reagent, the more blue that reagent turns. And so the degree of color change that you see is proportional to the concentration of the protein. And we can monitor this color change by measuring the absorption at 595 nanometers on a spectrum. So the spectrum will look a little bit like this. So the red line here is the color of the Bradford reagent, is the brown version of the Bradford reagent when it's not bound to protein. It absorbs really strongly at 465 nanometers, and it actually has a minimum here at 595 nanometers. On the other hand, the blue color of the Bradford reagent after it reacts with protein has a maximum here at 595. So when we look at the color change, especially the increase in absorption at the 595 mark, that's how we know that we have a higher concentration of protein in the sample. So how do we then relate the blue color that we measure with our protein of interest to an actual numerical calculation of concentration? We do that by using standards. So standards in this case refers to known concentrations of a protein called bovine serum albumin, which is a sort of generic, fairly globular and fairly symmetrical protein. So you can create concentrations where you know how much protein is in the sample, and then you can essentially measure how much color change you have at that known concentration. And then if you have an unknown concentration, you can use a measured color change to then sort of work backwards and estimate the actual concentration of protein in that sample. So let's look a little bit at what a standard curve might look like. So here we have on the x-axis our concentration of BSA, and we make that by using BSA in a concentrated stock solution of which we know that concentration, and then we prepare 5 different known concentrations of BSA, and then we measure the increase in the blue color on the y-axis, which is the absorbance at 595 nanometers. And within the range of the standards, this gives you a pretty linear standard curve. And so then let's look at how you might use this to measure a protein. So usually even just by looking at your sample in the cuvette, the color of the Bradford reagent can kind of help you visually place this particular sample as being a little bit less blue than this standard and a little bit more blue than that standard. It's somewhere in the middle of them. And the way this curve is mapped out, the most brown standard is down here at the lowest concentration. Our 5th bluest standard is up here. And so the 3rd and 4th ones where our unknown is between are at these two. And so our unknown probably is maybe about this blue in color, right? You can measure the absorbance at 595. It's somewhere between the 0.3 and the 0.4. And so in absorbance. And so once you can map that onto your standard curve, then you can use the y to determine what the value of x is, somewhere between probably 100 and 150 micrograms per milliliter. And so sometimes what can happen is you'll have a sample that is very, very, very blue, much more blue than the highest standard that you created for your standard curve. If that's the case, if you have a protein unknown sample that is way outside of your standard curve range of absorbances, you will not be able to get an accurate measurement of the concentration of that unknown sample. So what you'll need to do to be able to get a readable, predictable measurement is to dilute your sample, which is this point here. So if you have a sample that's very overly concentrated, you'll need to dilute it before you measure it. And then you'll just have to remeasure the absorbance at that lower concentration, and then you can multiply back to figure out your original concentration of your original standard. And we'll talk a little bit about more how to do those calculations in a little bit. And just as a reminder, the Bradford assay is going to give you the concentration of all of the proteins in a sample. The Bradford reagent doesn't distinguish between different proteins for the most part. So let's talk a little bit about how you'll actually run all of your samples. So first you'll run your standards, and to do this you'll prepare the standards in 1.5 ml tubes, which look like this. They're little microcentrifuge tubes that have a plastic cap that snaps on. And so in this case we'll be making about 500 microliters of your standard, which will be, you'll have plenty of space in the tube to do this. And then you'll use some of that standard. Of that 500 microliters, you'll use 100 microliters, and then you'll mix that with 2.5 milliliters of the Bradford reagent, and you'll do. So you'll place the cuvette in the UV-vis spectrophotometer, and this is what a cuvette looks like when it's in the spectrophotometer. The cuvette is basically a little rectangular container that you can fill with your standard or your sample, and then light is passed through your standard or your sample, and then the absorbance is read, and then you can use the absorbance measurement at your wavelength of interest to then figure out the first make the standard curve, which we're doing now, and then later to figure out the concentration of your unknown sample. So once you've mixed together the Bradford reagent and the standard in your cuvette, you'll want to mix them well. And you want to do this to ensure that the Bradford reagent can mix uniformly with the standard that you've added to it. And you can mix it in a couple of different ways. So you can use a micropipette, and you'll want to use a fairly large tip size and volume, probably closer to the 1 milliliter, to be able to mix thoroughly and efficiently. And if you don't want to actually pipette the sample up and down, you can also just stir in the cuvette with the pipette tip, and then that should be sufficient enough to mix it pretty thoroughly. And the third option that you have is to just take the cuvette and invert it a couple of times. But if you want to do it this way, don't just put your gloved finger on the open end of the cuvette and mix it. You don't want to get your anything on your glove, and you don't want to contaminate anything else that you touch with your glove. So if you want to do this, make sure you cover the top of the cuvette with parafilm. And the parafilm will keep the liquid inside of the cuvette and stop anything from, making a mess or contaminating anything else. So once you've done that, once you've mixed the Bradford reagent and the standard, you'll let the reaction occur for at least 5 minutes. If you end up waiting for longer than 5 minutes, that's completely okay, but make sure you wait at least 5 minutes. And then finally, you'll measure the absorbance spectrum using the SpectraVis Plus. And the SpectraVis Plus is just a little spectrophotometer that's going to be plugged into a laptop, and you'll receive more instructions about how to operate the software in the laptop. And so something that's really important to keep in mind when you place your cuvette into the little cuvette holder over here is that on all spectrophotometers, you'll see a little arrow, and the arrow indicates the path that the light takes through the cuvette. And so this doesn't matter too much if the cuvette you're using has 4 clear sides. The light can pass through either of the sets of edges. But some of the cuvettes, you might have frosted sides on 2 of the sides, or perhaps lines on 2 of the sides. And so if that's the case, you want to make sure that the light path is going through the clear sides of the cuvette. You don't want anything to get in the way of that light path. So finally, once you measure Once the SpectraViz takes the reading, it's actually going to generate a whole spectrum for you, and so you'll have to look through the list of absorbances at each wavelength and pick out the one that'll give you the absorbance closest to 595, and then you can absorb the absorbant. You can record the absorbance at that wavelength. So sometimes the spectrum won't give you a reading at exactly 595. You might get 594.8 or 595.1. So just pick the closest one that you can find when you're recording that absorbance. So running your samples will be very similar to running the standards, which we just talked about. So when you'll prepare the samples in a similar way. You'll mix together some of your Bradford reagent and some of your sample. You'll mix them well, and then you'll let them react for at least 5 minutes, and you'll measure the absorbance spectrum, and then you'll record the absorbance at 595 nanometers for each sample. So that mostly works pretty much the same. But here you might run into an issue if the absorbance that you read for your unknown sample is outside the range of your standards, especially if it's too high. So usually if it's above an absorbance of 1 optical density unit, then you'll want to dilute that original sample and then rerun the Bradford assay on your diluted sample. So if that's the case, you're going to want to dilute your original sample in one of those 1.5 milliliter tubes. And then you'll take 100 microliters of that diluted sample and mix it with 2.5 mils of the Bradford reagent in a cuvette. And then you'll do the same mix together, let the reaction happen, and measure the absorbance. And then if necessary, you'll have to repeat that dilution and run the Bradford assay another time. So let's now talk a little bit more about how to do those dilutions. So when you need to make a dilution, you don't want to take the solution that's in the cuvette and dilute that. You want to take your original sample and make the dilution from there. The solution that's in the cuvette is sample mixed together with Bradford agent. It's not pure sample. You want to make sure that you take some of your original sample when you make this dilution. So the first dilution that you'll make when your unknown protein concentration is too high is you'll make a 1 to 10 dilution. And that'll usually get you within the range of the standard curves. So the way you'll do that is you'll take 1/10 of your total final volume. That's where the name 1 to 10 dilution comes from. So in this case, we're making a 500 microliter total volume. 1/10 of that is 50 microliters. So you'll take 50 microliters from your unknown sample, and then you'll dilute that out to 450 microliters of water because you want to just add water to make up the difference between your 1/10 and your total volume. And again, we're making extra. We only need 100 microliters of it to actually use for our Bradford assay, but we always want to make a little bit extra because we can't. If we were to make only 100 microliters, we wouldn't be able to actually draw up and dispense 100 full microliters. Some of it would be stuck, for example, on the outside of the pipette tip or on the walls of the tube. So that's one reason you need a little bit of extra. And the other reason we make a little bit of extra is just in case you need some of this diluted sample to do further dilutions with, or you had to redo the assay for any reason. So once you've diluted your sample, you take those 100 microliters and you put it in a cuvette with the Bradford reagent, and you'll do your same, mix them together, let them react, and then measure the absorbance of 595. So once you take that next absorbance measurement, you will then see if your unknown sample has now fallen in the range of your standard curve, or if you need to potentially do a second dilution if your protein is still extremely concentrated. And a second dilution that you'll try is a 1 to 100 dilution. So the easiest way to do a 1 to 100 dilution is take 50 microliters from the 1 to 10 dilution that you just made. And then since again you're making 500 microliters total volume, you'll add 400 microliters of water. So what you've just done there is taken a 1/10 dilution of a 1/10 dilution. So multiply together the 2 10s, and that gives you what you now have is a 1 to 100 dilution. And then once you do the Bradford assay with your 1 to 100 dilution, that will almost always get you to a readable protein concentration. All right.